Hello, so we're back to talk about a serious and sad disease, which is AIDS, acquired immunodeficiency syndrome. So this disease started when in the United States you started to see clusters of patients developing two different conditions, which one was the pneumocystis pneumonia. Pneumocystis jirovecii is protozoa that causes pneumonia, usually in immunocompromised patients. And you also had Kaposi sarcoma, which is a type of cancer that usually has manifestations on the skin, that is also common in immunodeficiency patients. And they were trying to investigate where that immunodeficiency came from. And it was discovered in young homosexual men. So there was a trend here. In 1983, they discovered the HIV, which is the virus that causes AIDS, the human immunodeficiency virus. This virus has a selective ability to infect T helper cells because they possess a molecule that is called CD4. Also, they can infect macrophages and dendritic cells. Okay? Why? Because these other cells also have CD4. Okay? And then later on, as let me just try to change my slide here. Later on, when they started to study populations of chimpanzees from Central and West Africa, it was seen that those animals had a similar disease to the one that was called AIDS in humans. Okay? What happened is that there is a theory that humans were eating chimpanzee meat, and that's how they acquired. But this is a theory. So the HIV is from the genus lentivirus. It is a retrovirus, meaning what? Let's remember what a retrovirus is. It has an RNA with it, which is two identical single strands, and it can make from RNA DNA. And then this DNA can incorporate in the genome as a provirus. This is why it's so hard to get rid of this virus, right? Because once it's in the DNA of the host, that would be a bacterium. Let's say this is the cell, and this is the DNA. Once it incorporates, this is the provirus, right? In the DNA of the host, then it's practically impossible to get rid of it because it doesn't excise anymore. Okay? There is a very important spike protein. Remember how the spike, if this is the virus, this is the capsid, this is the DNA, the spike is like here, called GP120. And I'm going to tell you how GP120 is important in this process of selecting T cells, macrophages, and B cells to infect. So this is GP120 here, right? This is GP41, this part right here, okay? And these two together make the spike. The spike of the virus is able to connect to trying to move down, but it's not working for me. Okay, there we go. So this is again the virus, the GP120, the GP41, and how connection with the CCR5 or CXCR4 co-receptors and CD4 receptor from a CD4 T cell will allow the virus to enter the T cell. This is like the door, okay? This is like what you have in the T cell, which is this. Oops. This would be the door. Oops, my pen is not working. and whatever is in the spike, so the spike of the virus would be a key to enter that cell, okay? So once the virus is in, it fuses the viral envelope with the plasma membrane, and once it's in, and it's going to incorporate as a provirus in the DNA of the host cell, which is humans. This disease only affects humans, okay? So this is what happens in the T cells. They become latent by becoming a provirus, right? The provirus is right here inside the chromosomal DNA of the human cell, human T cell. And once there's a provirus, it can make multiple mRNAs, make the viral proteins. Usually, it always packs the viral RNA with the reverse transcriptase, which are these little blue dots here, which only the virus can make, right? We cannot make reverse transcriptase. They can make it, and they can make DNA from RNA. Okay. In macrophages, they can also be latent as a whole virion inside vacuoles, okay? Because remember, macrophages have a lot of vacuoles inside the cytoplasm. So that's another way that they become latent, okay? So before we talk about the stages, I want to mention to you that the presence of CCR5 is one thing that allows the virus to enter, because it's part of the door, right? It's part of the keyhole where the key is gonna get in. The thing is, some people do not have CCR5. And what does this make them? This make them resistant to HIV. People that do not get infected with HIV, a lot of the cases are because they do not have CCR5. Just wanted to make this point before we continue. So the disease has 3 stages. In the first stage, what you're going to have is the virus infects the cell. So then blue here is our viral count, blue plasma HIV RNA. So when you count RNA from the virus in your blood, in the beginning of the infection, which is the first 3 months or 6 months, you have a peak, okay? And that peak of RNA happens because the virus is multiplying. As it's multiplying, what's dropping? CD4 T cells. And then your immune system, as a way to compensate for this loss, starts to produce a lot of CD4 T cells. And then you have a peak of CD4 T cells. What is happening here? Remember how CD4 T cells are helpers, right? What do they help? They help to mature B cells, and they help to mature CD8 into cytotoxic T cells. What are these cells for? Well, B cells are going to make antibody anti-HIV. CD8 T cells are able to kill virus-infected cells. Okay? So any cell that they see with HIV, it kills, and the HIV is dying together before they can continue to proliferate. So with this increase in CD4 T cell, what you're actually doing is killing virus, because you're enabling CD8 T cells and B cells to destroy this virus. The virus count goes down considerably, and then you have enough CD4 T cell, right? It starts to steadily decline the amount of CD4 T cells. Why? Because this line here represents HIV particles that are already as a provirus in the blood, in the CD4 T cells, and these ones could not be eliminated. The CD8 population that is growing here because of that CD4 T cell peak is not going to be able to eliminate every single infected cell. So some of them remain, okay? Because if they're latent, they're not really visible. They're just quiet inside those CD4 T cells. So you have that low amount of virus in the blood for the whole course of the disease. So phase 1 ends with CD4 T cells count starting to decline, and then in this first time you're asymptomatic, or you only have a chronic lymphadenopathy, which is an increasing lymph nodes. This second phase is symptomatic, and you have early indications of your immune failure. So your immune system starts to decline because as the CD4 T cell population is declining, what's going to happen? You're not going to be able to do this, and you're not going to be able to do that, right? So you don't have the whole defense here being built for your immune system to be functional. And then in phase 3 is where you start to see more indications of immunodeficiency, which is the phase that we call AIDS. Okay, so this phase is AIDS. What do you have here? You have different types of signs and symptoms that include pneumonias caused by fungi, pneumonias caused by protozoa. You also have some types of tumors being formed. Let me show you this table. I have a table of some diseases that are associated with AIDS. So you see protozoa infection, viruses infection, herpes is one that happens often, tuberculosis is common, the pneumocystis pneumonia, histoplasma, which is a disseminated fungi infection, meningitis caused by cryptococcus, candida, which is causing oral and vaginal candidiasis, or what we call yeast infection, and some types of cancers. Okay, so these are common diseases. So what are the signs and symptoms of AIDS? The development of other diseases because your immune system is weak. Okay, so reason why it's so hard to make vaccines, because you have provirus inserted in the DNA. That's the main reason. Okay, second reason, it is harder to make a vaccine against viruses. Viruses can mutate. That makes it harder for you to have one vaccine that works well, right? For diagnosis, seroconversion is the period when the infection, between the infection appearance and the appearance of the antibodies. So you get infected, but you still don't see antibodies. And seroconversion is when you start to see antibodies. It takes up to 3 months, okay? You can detect HIV antibodies by ELISA or by Western blot, but if you really want to know how many viruses are in the plasma to determine what phase of the disease the person is, you need to do a PCR. And a PCR is going to amplify this viral RNA and quantify it, okay? Transmission happens because HIV survives 6 hours out of the cell. So even if it's not inside a cell, you can still get it from the sperm, the semen, even if it's not inside the cell. Routes of transmission: sexual, breast milk, transplacental infection of a fetus. Usually 40% of the fetuses of mothers that have AIDS can get infected. And this type of infection from mother to kid, to fetus, usually happens during birth. So they try to avoid vaginal birth because the volume of blood that goes to the placenta during the labor period, during that process, is really big. So the chances of the mom to infect the baby during birth is bigger than during the whole pregnancy. Okay? Blood contaminated needles, organ transplant, artificial insemination, and blood transfusion. Anal receptive intercourse is the most general form of sexual contact, which does not mean that homosexual people are the most affected. Look at this. Heterosexual transmission is the most common mode of HIV transmission. Okay, so there may be some misconceptions about this, where sometimes you could hear that homosexual would be the ones most affected, and that's not true. Okay, Most of the people infected with HIV are in Africa, even though this disease is in other continents like Eastern Europe, Central, South Asia, also happen a lot because of injected drugs. People don't change the needle and use drugs that they shouldn't be using, right? How do you prevent? Using condom, testing for HIV, using needles that are not contaminated, having behavioral intervention like sexual education where you learn that you should do it safe and, you know, not have multiple partners and use condoms and stuff like this. And like I mentioned to you, the challenges in developing the vaccine come from, you don't have a research animal model because this disease only happens in animals. In men, which are animals. There is not a full understanding of the mechanisms of retrovirus, high mutation rate, which leads to resistance. And remember, we always mention that it's much more convenient for industries, pharmaceutical industries, to make medicines that are super expensive and are used to treat the disease than to make a vaccine and then not sell those medicines anymore. The medicines that are used, so the drugs that interfere with the HIV life cycle, would be interfering with fusion and entry. You don't need to know these names, okay? I'm just mentioning how these drugs interfere. They can also interfere with reverse transcriptase and inhibit the enzyme, so they cannot make the DNA. There are drugs that interfere with integrase activity, so they can integrate the DNA, and there are protease inhibitors. There are a lot of proteins that are enzymes in the virus that are proenzymes. So once they are broken down into a smaller protein, then they're active. If you break the ability of these enzymes to break down these other enzymes that are the proteases, so proteases break down proenzymes into active enzymes and then they activate them. If you don't have that ability of protease activity, the molecules in the viruses are not going to be active and then the virus cannot survive. So that's another way of interfering with their life cycle. And with this, I am done telling you what you should know about HIV and AIDS, and let me know if you had questions.